Welcome to our presentation on the process of meiosis. This is a review. Key concept here is during meiosis, diploid cells undergo two cell divisions that result in haploid cells. These haploid cells are needed for sexual reproduction. They have half the number of chromosomes that a normal diploid cell has. In your body, you have trillions of diploid cells with both sets of homologous chromosomes. Only in your sex cells, sperm or egg, do you have haploid cells with half the number of chromosomes in them, in preparation for joining with another gamete so that the two halves make a whole, a new individual with the diploid number. In the process of meiosis, cells go through two rounds of cell division. Meiosis reduces the chromosome number and creates genetic diversity. Meiosis produces genetically unique cells, not identical like mitosis, but unique, different cells, results in the haploid number, and takes place only in certain times of the cell's life cycle, and it's the process that involves preparation for sexual reproduction. Here we have 4 chromosomes. They've all been replicated. Each has 2 chromatids in them. And when we go through the first division of meiosis at metaphase and anaphase, the homologous chromosomes are separated. And then the second division in meiosis separates the chromatids, and where we started with 4 chromosomes, we now have 2 in each cell. We have 4 cells with 2 chromosomes in each. It's unique and different, and even the possibility of crossing over can increase the genetic variation. Meiosis 1 and 2 each has 4 phases, similar to those in mitosis. Pairs of homologous chromosomes separate in meiosis 1. That's very different than mitosis. These homologous chromosomes come together at metaphase 1, all wrapped around each other, not lined up one after each other, but the homologous chromosomes are all wrapped up. These homologous chromosomes, remember, are similar. They have the same kinds of genes on them, eye color, hair color, but they're not identical. You may have a gene for brown hair, you may have a gene for black hair, you could have a gene for blonde hair. They're different genes on those sites, but each of them has the same combinations of genes, eye color, hair color, things that we can see. And don't forget, there are millions of genes that we can't see that make the enzymes that keep us alive. We refer to the genes that we can see the results of, eye color, hair color, things like that, curly hair, straight hair. But there are way, way more genes that keep us alive and keep us going that we never see the expression of on a daily basis. The sister chromatids divide in meiosis 2. So when we go to the second division, the X's do line up very much like mitosis, and the spindle fibers pull the chromatids apart, giving us 4 cells with half the number of chromosomes. Sister chromatids are copies of the same chromosome. Here we have a homologous chromosome, 2 sister chromatids on each, and they'll be separated, and there'd be 1 chromosome. Meiosis 1 occurs after the DNA is replicated. Whether it's mitosis or meiosis, the DNA replicates in the S1 phase of the cell cycle. So this is all, in both cases, it's previous to this step, to cell division. Meiosis 1 divides homologous chromosomes in 4 phases. Prophase, the chromosomes form, the nuclear membrane dissolves, and centromeres start sending out spindle fibers. But here's the key: in metaphase 1, the homologous chromosomes line up together, wrapped up at the equator, not all one after each other. Anaphase 2, the homologous chromosomes, not chromatids, chromosomes are separated. And then in the telophase, the nuclear membrane forms again, and the chromosomes are separated, and we have cytokinesis. The cell divides. This is the part where the reduction number takes place. We started with 4 chromosomes in each, and now we have 2 in each. Metaphase 2, just very much like mitosis, just divides the chromatids, the sister chromatids, into 4 separate cells. Again, the DNA is not replicated, it already has been, so there's no DNA replication that happens in the S cycle of the cell cycle. And here we have the pickup from the last. We've got 2 cells with 2 chromosomes in them, but they're still X's. They all line up now. It looks more like mitosis in metaphase 2. And then in anaphase, the chromatids are pulled apart, and we end up with 4 cells with 2 chromosomes in, when we originally started with 1 cell with 4 chromosomes in each. Meiosis differs from mitosis in a number of significant ways. Meiosis has 2 cell divisions, mitosis has 1. In mitosis, homologous chromosomes never pair up. In meiosis, results in haploids. Mitosis results in diploid cells. Here we have mitosis, produces genetically identical cells, results in a diploid to a diploid, takes place throughout the organism's lives. Your skin is reproducing this way constantly, and it's involved in asexual reproduction. In meiosis, produces genetically unique cells, different cells, results in haploid cells, takes place only in times when reproduction occurs, and it sets the table for sexual reproduction. Haploid cells develop into mature gametes. Gametogenesis is production of gametes. Gametes differ from male and female. Sperm becomes streamlined and motile, which means movable. Sperm primarily contributes to DNA to an embryo. So here we have, from the germ cell, the diploid cell, we go through the 2 divisions, and we end up with 4 sperm cells, smaller, about a quarter of the size of the original, because the cytoplasm is shared, and the little tails allow them to swim around. That makes them motile. Eggs contribute DNA, cytoplasm, and organelles to the embryo. So the sperm really only contributes the nucleus, its haploid nucleus. The egg has to contribute cytoplasm and organelles to the embryo. During meiosis, the egg gets most of the contents, and the other cells form polar bodies. So what we have here is a single germ cell. It divides. One of the cell in the first division gets almost all of the cytoplasm, organelles, and everything. The other polar body just gets basically a nucleus. And then when it undergoes a second division, again, we have a large egg, a small polar body, and actually the second polar body here from the first division will also divide again, so we'll end up with 1 egg and 3 polar bodies when we started with 1. So the egg gets most of the cytoplasm, organelles, the good stuff that a cell has, and the 3 polar bodies end up with just a nucleus and aren't used for anything. Except in plants, sometimes in the fruit, like an apple, an orange, the polar bodies will be used to create the endosperm, and that is what the apple that we like to eat is, and the orange and the grapefruit. Grapes, the thing we like in a fruit, is actually the endosperm, which is created from the polar bodies.